Yeah, so first I want to thank the organizers for an amazing schedule today and all the speakers. It's been an amazing set of talk and I've really enjoyed it. And then I'm very grateful for this opportunity to come and talk about some of the research that I'm doing. So I will talk about hybrid speciation in birds. And as most biologists, I'm fascinated by the amazing set of adaptations and species that have been arising out in nature. And the main research interest that I have is how the variation that selection can act on to achieve this amazing variation originates. And there are a few processes. So of course, new mutations are the best known ones. But there's been, and they can then of course fix by selection or drift. But as Justin was showing on this little scary, for instance, sorting of standing genetic variation has been shown to have power or have a potential to lead to rapid adaptation. And then, as we started to sequence entire genomes or larger parts of genomes, we have increasingly realized that new combinations of old variants through hybridization can lead to new diversity. And today I will talk about the potential of hybridization to lead to novel variation. And I will start by introducing hybridization and how it can contribute to novel variation. Maybe it's going to be a little bit basic level now that I see the select audience here, but bear with me. And then I'm going to go into how divergent genomes can combine and create novel variation using the research that I and others in the sparrow system have been doing. And finally, I'm going to talk about something that I'm excited about getting input on, how altered regulation of gene expression could potentially enable novelty from hybridization. So I'll start about talk about hybridization, because many non-biologists might think about this when I say hybrid, and maybe they will think, but aren't they sterile? And this is quite along the line of thinking of some of the influential architects of the modern synthesis. So Fisher has said that one of the grossest blunders in sexual preference we can conceive of an animal making is hybridization, right? And generally, many hybrids are sterile. So there's, for instance, only 60 documented cases of fertile mules since the 1500s. But these few mules can enable a trickle of gene flow that can actually transfer some adaptive variants. And as we've been starting to use those novel sequencing methods that have enabled us to sequence entire genomes, we have seen that hybridization is common, not only in plants that was realized early on, but also in animals. And again, we started to look into different parts of genomes, and then we realized that different parts of genomes could have different evolutionary histories, and that species trees are more reticulated or web-like as previously thought. And as we heard in many of the talks today, new combinations of old variants have been shown to enable rapid adaptation and speciation. So why study hybridization-derived variation? Well, in some systems and for some traits, it's been shown to be an important source of novel variation that selection can act on. For instance, there's a study by Peter and Rosemary Grant in the 90s for the Galapagos finches, where they showed that beak variation was originating from hybridization to a high extent in some systems. And there's also evidence for new lineages arising from hybridization. Here again, an excellent example in Galapagos finches where GSP 40 is hybridized with Conorostris and gave rise to this new lineage. And hybridization has been shown to be at the root at some of the most amazing adaptive radiations that we know of. For instance, this adaptive radiation of cichlids in the Lake Victoria arose from an initial hybridization event. And we don't even need to go beyond this room to see that organisms with hybrid histories thrive. And understanding hybridization-derived variation can be important, because even small amounts of hybrid variation can have large fitness effects. So our inner Neanderthals actually affect our COVID prognosis. So if you get the nature allele, that's bad news, but the PNAS allele is going to give you an easier time. And variation from hybridization differs to that from mutation, because whereas mutation is typically point mutation, hybridization can transfer large co-adapted gene complexes or genes that have many changes that work well together. And hybridization or mutation is random, right? Usually negative. But hybridization transfers variation that's been tested by selection in a genomic background that's similar to its own background. And hybridization has also been shown to give rise to different types of evolutionary responses, including, for instance, hybrid vigor and extreme phenotypes. But to take a step back, sorry, I'll go through this quickly, but hybridization, depending on the setting and the organisms involved, it can have different outcomes, of course, ranging from absolutely nothing. If reproductive isolation is total, it's just a waste of gametes. If there's no reproductive barriers, they can all merge into one taxa. One of the organisms is more common, of course. You can swamp the genome of the other. Hybrid zones transiting between two parental forms can arise, as in Justin's example, and there are adaptive outcomes as well. For instance, when initial hybridization leads to full hybrid and then backcrossing continues until only the adaptive alleles from the other parental background persist, so adaptive introgression. But what I will talk about today is hybrid speciation, when hybridization give rise to a new lineage. And hybrid speciation is different from bifurcating speciation in that the two lineages that create this hybrid lineage already have some reproductive isolation. And for hybrid speciation to occur, it's absolutely crucial that the hybrid taxa achieves reproductive isolation against both parent species, at least if they're in symmetry, because otherwise, of course, they would just be a hybrid swarm again. And it comes in two flavors, one with no increase in chromosome number and subsequent recombination forming the genomes, homoploid hybrid speciation, and then there's allopolyploid hybrid speciation with an increase in chromosome number. And I'll talk about the first case here today. And I'm going to use this Italian sparrow system to address how divergent genomes can combine and generate new variation. And I'm interested in constraints and so forth in this. And the Italian sparrow system has been developed by Professor Glenn-Peter Sætre at the University of Oslo. So this is work I started as a postdoc in Glenn-Peter Sætre's lab. And I've since been developing the island system of Italian sparrows as a project. So the Italian sparrow is the middle one here. It's intermediate between its parent species, with the head being more Spanish sparrow-like and the body a little bit more house sparrow-like. It's also intermediate at the genomic level, and most of you will recognize these figures. A bar that's fully blue corresponds to entirely house sparrow-like, fully red, entirely Spanish sparrow-like. The Italian sparrow at mainland Italy has an intermediate genome. And the parent species are isolated by some approximately 0.8 million years. The house sparrow evolved in the Middle East when agriculture started. And then it recolonized the Mediterranean and met Spanish sparrows approximately 5,000 years ago. So I used the system to address whether parental genomes can combine in different ways. And why would we expect parental genomes to combine in different ways in this system? Well, because they occur on a set of different Mediterranean islands, and they look slightly different on these Mediterranean islands. One hypothesis would be that the house sparrows have just accompanied humans as they expanded with agriculture throughout Europe, just as they have accompanied humans approximately to everywhere else. The house sparrow is the most invasive bird species in the world. It's very adaptable. So we set out to test these hypotheses. We sequenced whole genomes of entire populations of individuals from Corsica, Sicily, Crete, and Malta. And we also sequenced these parental species in blue and red. And this is a principal component analysis. It basically shows that the main axis of variation in this whole genome data set is between the house sparrow and the Spanish sparrow. Better not use that one. Oh no. So what you can also see is that those Italian sparrow populations are basically lining up along this axis of divergence between those parental species. And you can see this even more clearly if we use one of these clustering analyses, where the Spanish sparrow is entirely red again, house sparrow is entirely blue. So the proportion of these parental genomes in the Italian sparrow vary between approximately 75% Italian sparrow and 75% house sparrow. And the only two that have similar proportions of the parental genomes are Malta and Sicily. But if we divide the ancestry analysis per chromosome instead, so this figure has entirely House on the top and entirely Spanish on the bottom. So the lower the curve, the more Spanish sparrow-like. And as you can see, the Maltese population is very different from the Sicilian, for instance, on chromosome 2. So not quite similar. And we wanted to understand whether different parts of the parental genomes were inherited by different hybrid populations. So we chopped up the genomes in very small pieces, or some 100 kb windows, and performed independent phylogenetic analyses of these windows. And the gray blur you see here in the background is all corresponding to a specific phylogeny that occurred in at least one of these analyses. And the thicker black line is not the true, it's just the most common phylogenetic tree. So definitely there's different Italian sparrow populations have inherited different parts of their genomes from different parent species. So yes, we have different regions of the parental genomes and different proportions of their parental genomes leading to hybrids. But aren't there any constraints on hybrid genome formation then? We would expect there to be constraints on hybrid genome formation because, of course, if we have an original lineage that then splits up in two, there can be this derived allele, and this derived allele, if net neutral or like advantageous, it can fix. And in another population, another allele can fix. And when they get together, of course, they might not always be compatible. Maybe the two proteins folded in different ways and can't work together or something like that. So hybrid species basically must unfit the recombination variants that are not fit. So do we see any evidence for such incompatibilities in the Italian sparrow? Well, we set out to cross the parental species again to try to understand whether there were such incompatibilities present. And what you see to the left is a picture of a normal ovary on the top row and an atrophied ovary in the bottom row. And half of the hybrid females had those atrophied ovaries. So we do know that there's some pretty detrimental variation segregating. But again, the Italian sparrow is not the first generation hybrid. They have been formed approximately 5,000 years ago, and there's been selection acting on these genomes. So can we detect any signs of what would have been incompatible populations or combinations in those genomes? So now I'm showing a little bit of a different figure. So this is a sliding window, like clustering analysis of the Z chromosomes, starting from the first to the last position, and entirely blue means that all the individuals in the population have a house sparrow allele for this part of the Z chromosome. So as you see here, this is the Cretan population. It has a high proportion of house sparrow DNA. But if we look across the other populations, even the Maltese and the Sicilian populations that have high proportions of Spanish sparrow DNA have some regions of the genome, as you can see here, furthest to the right, and other places on the Z chromosome that are very house sparrow-like also in these populations that have high proportions of the Spanish sparrow genome. And in those regions, we find strong enrichment for DNA repair genes and mitonuclear genes. So mitonuclear genes are important because mitochondria and mitonuclear genes need to work well together in order for respiration to work very well. And here's a mitochondrial network, and the blue, dark blue here is house sparrow, the red is Spanish sparrow. The length of these branches correspond to the number of differences between the mitochondrial haplotypes. And as you can see, almost all of the individuals of Italian sparrow have the house sparrow or house sparrow-like haplotypes. We see one individual that has a Spanish sparrow mitochondria, and two of the Maltese individuals have both of the parental variants. So it's not that they have variants that are like intermediate, it's just how it's been drawn. There's no like software for drawing heteroplasmy in an efficient way to my knowledge. If there is, please tell me. But basically heteroplasmy is one of these phenomena that you expect to arise from incompatibilities. So one of the things that I'm excited in to look into further, and that we're now starting up, is to look at how the proportion of the genome that's inherited by the house sparrow affects the efficiency of respiration, because it should work better together with the house sparrow mitochondrial genome. Okay, we have many different combinations of parental alleles. They are constrained to some extent because we don't want incompatibilities. So can intermediate phenotypes arise or non-intermediate phenotypes arise? So hybrid species have this increased variation that we expect to be able to that we expect could potentially be able to serve as substrate for selection and lead to novelty. And I was interested in whether it could lead to entirely novel non-intermediate morphologies and whether the variation could be used to adapt locally. So we looked at a bunch of Italian sparrow populations, some on mainland Italy, here denoted in green on the map to the left, and some island populations, and these are denoted in yellow. Again, we have the Spanish sparrow in red, a house sparrow in blue, and you can see that these line up along the continuum of Spanish to house sparrow. And we quantified a few traits that differ between those species, including some plumage traits, such as the crown and back, color. We also looked at beak size and shape. And we quantified coloration in a multivariate way to get the variation there and not just the mean in the color. So we created some color PCs. This is work by Homa Papaliasti. He's a former postdoc in the lab who did an amazing work with this. And we made this main axis of variation for each of these traits. And as you can see, there are different patterns for different traits. For instance, here, beak shape. We have the house sparrow at one end, the Spanish sparrow at the other end, and the Italian sparrow is intermediate. So for some traits, we have island populations being more house sparrow-like and Spanish sparrow populations more Spanish sparrow-like. It's also the case for rump. But I think it's most exciting with, for instance, the beak size, because here we see this pattern where the Italian sparrow actually has a more or a larger overall beak size than both parent species. And we also see relatively large variation in beak size between those populations. So I was wondering, is this something that could be adaptive? So if we would expect beak differences to be adaptive, we would think that the variation in beak size and shape should be best explained by some environmental variable, rather than with genomic contingencies. And we sampled 3 populations on each of these islands. Those like colorful drawings are the beak shape outlines for for these island populations. So we looked into the effects of, or like to which extent the variation in beak shape and size was explained by climate variables, diet variables, and genomic composition. And what we find is that beak size is best explained by a temperature seasonality at the population level. So these points all correspond to one population each. And temperature seasonality basically reflects the temperature in the hottest month during the year. And like the hotter the temperature, the larger the beak size. And this is, again, just a tiger in the grass kind of situation, right? We haven't been testing this experimentally, but it could be that it's important to be able to thermoregulate on these arid Mediterranean islands. Beak shape, on the other hand, was best explained by the delta C13 value in the diet. The delta C13 value is lower in C3 plants, and these are just as house sparrows, pretty much all Italian sparrow populations are very anthropogenic. So it would also be interesting to look into whether these that have the beak sizes that are correspond to lower delta C13 values would be better at cracking larger, harder seeds, because delta C13 values are lower in like those cultivated plants. And interestingly, we also find that the 2 populations that have the largest difference in beak shape, the Sicilian and the Crete one, among the outlier loci that are most divergent between those populations, we find one gene called FGF10. And on this FGF10 gene has previously been found in Darwin's finches to be associated with beaks. So it would be really interesting to look at like the expression of this gene and how much is expressed and whatnot in those two populations that have different beaks. But this is also to be confirmed experimentally. And then finally, I'm going to talk a bit about altered regulation and of gene expression and a funky idea about how this could potentially enable novelty from hybridization. So hybrids have long been known to give rise to non-intermediate phenotypes. And here I'd illustrate this by a larger green bird that's bigger than both parent species. But it could also be the example of the beak size that was extreme in the case of our Italian sparrows, right? And a very nice example of this is, of course, the Lauren Rieseberg's lab's work on this Helianthus anomalus that has a transgressive ecophysiology that enables it to inhabit habitats that are drier than both parent species. And the traditional explanation to transgression has been this complementary gene action, where there's plus alleles for here stature or for like plant height in species 1 on the three out of the 5 genes, and in species 2 on the other 2. And then, of course, we can have transgressive sorting of these plus alleles. But the question is whether these alleles are for should be pluses and minuses for coding variants, or whether there could be regulatory elements that these could denote. Because I think the idea that intermediate genomes give rise to transgressive phenotypes a little bit begs the question about asking what happens to regulation of gene expression in the middle. So Homa and I set out to look at the gene expression data set for the testes here. And what we would expect if we have an intermediate genome that has parental alleles from 2 species would be that it would be within the range of the parental alleles or parental expression typically. And this is denoted here by the yellow dots. That's within the range of the parental species. But we also find genes that are more expressed than in both parent species. You see it has a high value of this Italian to house sparrow ratio and a high value of Italian to Spanish sparrow ratio. So they are like transgressively overexpressed. And we also have genes that are less expressed in the hybrid than in both parent species. And this was 22% of the genes, so that's pretty many of them. And in general, what we know about hybrid incompatibilities, metabolism, and so forth makes sense in terms of what we found being overexpressed. So, for instance, we found genes involved in the mitochondrial respiratory chain, ribosomal proteins, and so forth, cytosolic ribosomal proteins, and like basically the overexpression of these genes could potentially be some kind of solution to this mitonuclear incompatibilities that we see reflected in other studies in the genome as well. We also find that there's overexpression of genes involved in the binding to the sperm to the zona pellucida, and that's when the sperm like fertilizes the ovary. This needs to happen. And this is a function that has been shown to be under selection in a sperm protein study by Melissa Ray. Okay, so getting back to this data again, I'm going to show this in a bit of a different representation. So on the top panel here, we have the different differences in expression between the Italian sparrow and the house sparrow. And as you can see, those yellow dots that correspond to differentially expressed genes, there's pretty many of them. And there's also pretty many of these differentially expressed genes at the bottom panel. And that corresponds to the difference to the Spanish sparrow. So approximately equally many genes are differentially expressed compared to the house sparrow and the Spanish sparrow if we look at the testes. But if we instead look at the ovary, the expression is very house sparrow-like, with very few of the genes being differentially expressed in comparison to the house sparrow. But many genes are being differentially expressed in comparison to the Spanish sparrow. So we need to understand what's the basis of novel hybrid gene expression. And this has been much more studied in allopolyploids, the ones that have like genome doubling and so forth. because we understand that there's been subgenome dominance and so forth. But I think that, very simplified, of course, regulation of gene expression is an interaction between different regulatory elements. So extremely simplified, there are these cis-regulatory elements that are close to the gene. There are also trans-regulatory elements that can be found in other places in the genome. And together, the combination of these decide how much gene product or transcripts are going to be produced, right? And it's generally under stabilizing selection. So if there's a change in transregulatory gene transregulation, this is typically opposing this regulatory change. And this might mean that there could be different combinations of these regulatory elements that can give rise to approximately the same amount of gene expression. And this possibility I've illustrated here as minus alleles in trans for this blue parent species, decreasing expression, plus alleles in this increasing it. And in the red example, it's the other way around, right? Plus alleles that would increase it in trans and minus alleles that would reduce it. And then, of course, we can combine these alleles in an F1 hybrid intermediate combination of alleles. And they could then again recombine in a transgressive way in second and further generations, giving rise maybe to more expression or less expression. This is just a wild idea, but I think it's an interesting one because we are expecting a decoupling of different parts of the genomes in hybrids, and this could very well go for the regulatory elements. So does it matter whether we think about complementary gene action as coding genes or as regulatory elements? I think it does. Because gene expression or the expression of one gene can regulate how much product comes out of an entire network and so forth. But it's an open question. And there's a bunch of other phenomena that happens in genomes that are also happening to a higher extent in hybrids. For instance, there's structural changes. If there's differences in the configuration of the genome structure of the parent species, we could expect inversions, deletions, insertions. There's been empirical data showing that methylation can be different in hybrids in Louis Bernachet's whitefish research, and transposable elements may also be increased in number, and their expression can increase, or they can be reactivated in hybrids. So potentially this could also lead to funky new patterns of gene expression in hybrids. So understanding this is something that I'm very interested in. So right now in my lab, we are setting out to use all of these Italian sparrow populations that have different genomes and try to understand what's been happening. How has gene expression evolved in this? Like, what are the general patterns of gene expression evolution in hybrids across tissues and so forth? Can we predict what genes are going to be differentially or transgressively expressed based on the networks they belong to, or what functions they have, or whether they are on the cis-regulatory or trans-regulatory or cis-trans compensatory regulation and so forth? And also, like, what are the roles of these different hybrid genome alterations, like structural changes and so forth? So that's what I'm interested in at the moment. So just to conclude what I've been talking about, hybridization can form many novel combinations of genes, enabling adaptation, but there can be some constraints to how these genomes can form, of course. The hybridization and the hybrid genomes can enable novel morphologies, and these can sort in adaptive ways. And hybridization can also potentially give rise to novel patterns of gene expression, but this is research that remains to be done. So thank you so much for listening. This is work that has been done by a lot of people. I want to thank the Sparrow crew with Glenn Peter Sætre and everybody else I was working with at the University of Oslo. My former postdoc, Homa Papoli-Yasti, who's been a queen of these bioinformatical analyses, the current hybrid express team that are in the field and working hard to start making this research I was talking about as future directions happening. My lab and the space lab, that's my home at Lund University, and a lot of funding. The Crafoord Foundation has actually been giving me money in the past, not for the Sparrow project in particular, but I'm still very grateful. So thank you so much for listening today.